Well, in Lab19, sheep brain dissection. So in this lab, we're supposed to dissect the sheep's brain. So I'm going to show you some sheep's brain picture, and you are responsible for knowing those big structure. So that's the sheep brain, and outside covered by meninges. And meninges have 3 layers: the dura, arachnoid, and the pia. Pia touch the brain, and dura touch the outside. And now we would remove those meninges. It look like this. So you found the sheep's brain does not have too much wrinkle. So it tell you not a lot of neuron inside. It's pretty lame. That's the cerebrum and 2 hemisphere, and those wrinkles, gyrus, sulcus. That's the cerebellum, and that's the spinal cord. So that's the top view. At the bottom view, you can see some structure. These 2 are actually olfactory bulb. They remove it. You should have these 2 olfactory bulbs. This is the cranial nerve number 2. So that's the optic nerve. When they combine together, this close. That's the optic chiasm. So you have the midbrain, the pons, medulla oblongata. So when we cut it open, let's do a sagittal cut. You are able to see all this structure. So let's look at the sagittal cut. This structure, the Y structure, that's the superhighway for left brain and right brain talk together. That's the corpus callosum. And when you dig in, you can find the lateral ventricle, but we don't move in. Let's look at the other structure. Okay, this is thalamus. Hypothalamus is around here. So the sheep, not like human stand straight. Sheep is flat. So their brain structure is like this. And this is the brain stem. So the midbrain, the pons protruding out. So this is the pons, and this is the medulla oblongata. And this is the cerebellum and the tree of wisdom, the arbor vitae. So this is arbor vitae. Let's look at the ventricle. The third ventricle is not very obvious. It's around the dicephalon area, but you can see the fourth ventricle. It will go down to here. So you found this is a small hole between the cerebellum and the brainstem. This is the fourth ventricle. So it will keep going down to the spinal cord to here. So that's basically the structure of the sheep's brain. And let's keep going to look at the Lab20. So Lab20 is the spinal cord and peripheral nervous system. So in this lab, if you have the photo atlas, you go to this page. It's pretty good structure picture to tell you the spinal cord. If not, you don't have it, you can use your textbook. So the spinal cord, okay, I take this picture from the book. You have 3 horns. You have the ventral horn, the dorsal horn, and the lateral horn. Actually, we use the big name. So the lateral gray horn, the ventral gray horn, the dorsal gray horn. So first you identify, okay, this is dorsal or ventral. So the butterfly is not completely symmetric. The gray area, if you connect to the side, and that's the dorsal. And the ventral, this is the ventral. It does not connect out. There's a white area here. And funiculi, this is those white structures. So you have the dorsal, ventral, lateral, funiculi, and you have 3 horns, the gray horns. And this one is called the ventral median fissure, and this is called the dorsal median sulcus. And the spinal cord, they're covered by meninges as well. So still 3 layers. The pia touch the nervous tissue. The arachnoid is between, and the third layer is the dura layer. So they have 3 layers. And this is the model we have in the lab. I can point at this, ask you, okay, what's this, what's that, like this. This will be the ventral gray horn, right? And let's look at this area. So this is your spinal cord. Is covered by 3 layers. The pia is this one. Arachnoid, this is arachnoid, subarachnoid space, and the outside is dura. So this is dura. And dura, between dura and the vertebra, you found this space is completely covered by fat. That's pretty normal in the spinal cord. You use connective tissue to fill the space. So this area with fat, this is epidural space. So when we talk about epidural anesthesia, is we put a needle here, and put the local anesthesia around here. So this is the epidural space. It's covered by fat. And the other structures are pretty obvious. And in the spinal cord slice, it looks like this. So this time it won't tell you which one is dorsal, which one is ventral, and you need to use the structure to identify. So when you look at the structure, you know, oh, that's the dorsal part, and the horn is sharp, and the ventral part, this is the ventral part. And also we're going to look at the plexi. So in the body we have 4 plexi. And these plexi are the spinal nerves that crisscross together. So we have the cervical, we have the brachial with the lumbar and the sacral plexus. So the cervical is around here, and the brachial, in the class I focus about the brachial plexus, is around here. They crisscross together, becomes 3 nerves, the median nerve, the ulnar nerve, and the radial nerve. They go to the arm. And the lumbar plexus, that's around here. So the lumbar plexus will crisscross, create a nerve called the femoral nerve. It goes to the thigh area. And the last one, the sacrum plexus. The last one is the sacrum plexus. And this around this area. The crisscross create a big nerve called sciatic nerve. It will go under the gluteus maximus and will go through the whole leg and go down. So these are the nerves and plexus. Okay, that's it.